This lecture is about cells. So I base many of my lecture slides from the free open text Anatomy and Physiology, and also from this Human Anatomy textbook. So let's talk about cells. And as we know, cells are the smallest unit of living. So a human being is a multi-billion, like trillions of cells in our body. And if we just look into one cell, we will see the membrane, which is the margin of the cell and lining of the cell, which we call cell membrane or plasma membrane, and cytoplasm, that is the space inside the plasma membrane. And there is one important structure in the cytoplasm, which is what we call nucleus. So we plan to gently explore each and every areas of interest in a cell. So now, when we talk about cell, this is what you see. This is the generalized structure of cell. And now you can see the outermost, which is the boundary covering, which is like a fence of a cell. That is what we call plasma membrane or cell membrane. And inside the plasma membrane, we have the space, which is filled with multiple contents. And this space is called cytoplasm. And cytoplasm is filled with the fluidary structure, which we call cytosol. And in the cytosols, there are many organelles floating around, including the nucleus. So now you can see the big nucleus here. In the center of the nucleus, we have a structure, little baby nucleus, which is called nucleolus. And around the nucleus, you can see the endoplasmic reticulum, to be more precise, rough endoplasmic reticulum surrounding there. And you will see mitochondria, which is the energy house for a cell. And you will see Golgi apparatus. And you will see like little elements running from here and there, like giving us a beam, which is giving the some degree of structural support. They are called cytoskeleton elements. So who start talking about cell? Who give a name cell? So this is the British scientist, Robert Hooke. He is from Isle of Wight, which is the little island on the west of the British Isles. And that was in 1635, around that time. He was using a magnifying glass and looking at a cork of a wine bottle. And if you look carefully with the magnification glass, then you will see many little small pockets or small little rooms in the cork. And this word came from Latin, which is cellular, which means small little room. And since then, the name, these small little pockets are called small little rooms, and then the term we got cellular. And now, with time, the scientists, and these two scientists from Germany, they start talking about human beings or any livings are made of multiple cells. These are the biological scientists. So when we think about cells, this is what we see in our eyes, like multiple cells, a human being, multi-billion, trillions of cells in our body, and they will be like that. And every cell has their own activity and function and doing their own job. This is something when it comes to our mind when we talk about cells. But if we look a little bit more carefully, it might be like that. multiple cells, one cell here, another cell here, another cell here, another cell here, many cells inside them, they will have all these. And it might be like that, but in reality, an individual cell has different shapes and different function. And if you look at the fat cell, it will be just like a little droplet of fat. If you look at macrophage with the loss of spike state, if you look at a nerve cell, it will be like that. If you look at sperm cell, the shape is like that. So what we see from these slides and what we see, what we have in reality, they will be different. Fibroblast cell will be like that. Erythrocyte, red blood cell will be like that. Epithelial cell might be like that, and so on. And when we talk about trillions of cells in our body, which cells contribute the most? Of course, red blood cell, RBC. In other words, erythrocyte, red cell. That contributes about 84% of cells in our body. If we think about neurons or neuroglia cell, it is 0.6% of the cells in our body. Have a little look about this diagram, which I think is very interesting. So for me, like this is what I see as cells. Like I have one cell here, another cell here, another cell here. This is the cell membrane or cell wall or plasma membrane. And this is inside the cell, cytoplasm. And these are the organelles inside the cell. So in between cells, there are small little space. That is what we call interspatial space. And inside the cell, we will call intracellular, and outside the cell, we will call it extracellular. This is my rough drawing about the cells. But of course, we have to use the microscope, do the histology slide, and have a look, put it on, use a high power beam, and we are going to see the cells we saw in our previous slides. So now cells is the smallest living unit of our body. Organelles, these are little organs which carry out essential function of the cells. And as we saw from previous diagram, we will say cells have three main components, which are plasma membrane, the outer boundary, cytoplasm inside the plasma membrane, which contain all the organelles, and the nucleus, which is the control center of cellular activity, which houses the DNA. So now let's look at the plasma membrane. Plasma membrane is a phospholipid bilayer. So we can see one phospholipid here with the hydrophobic and facing outwards, and hydrophilic and facing inwards, and another phospholipid with the lipid-like water-hate, hydrophilic and facing inward, and hydrophilic and facing inwards. And this is the bilayer, lipid bilayer, phospholipid. And on top of that, we will have protein here. Like this protein is what we call integral protein because it is embedded in the phospholipid bilayer. And there will be a few other proteins like which are just stacked on the cell membrane. For example, it is something like that. It's just stacked. It is not crossing the plasma membrane. Another one here, it is not crossing the plasma membrane. It is just stacked. So we will call these peripheral protein, and those embedded in the phospholipid we call integral protein. And on top of that, we will have cholesterol here. Look at that, cholesterol here, yellow bits in the cell membrane. So which is something like a lipid layer, phospholipid layer with mosaic of protein and cholesterol. And therefore we call plasma membrane fluid mosaic model because the cholesterol and phospholipid make the plasma membrane soft and liquidy. So the plasma membrane functions as the interface of interior and exterior. It provides the barrier against in and out of the cell. It also determines which substance from the cell inside the cell leave the outside and which substance from outside can come into the cell. And there will be plasma membrane proteins, which will also act receptors. So it is a very important structure of a cell. It is the boundary. As we say, plasma membrane has fluid mosaic model. Plasma membrane contains of mobile proteins embedded in lipid bilayer, and types of membrane proteins are integral protein and peripheral protein. We know that already. So membrane transport. How do we transport all the internal and external ingredients of the cell across this cell membrane? There are mainly three types. Number one is simple diffusion. As we know, the plasma membrane is structurally lipid bilayer, phospholipid bilayer, with the hydrophobic ends facing outwards, hydrophobic ends facing inwards, and hydrophilic ends facing inwards, and hydrophilic ends in here inside, which mean those substances which are lipid soluble will be easily able to just pass through the lipid-like structure across the cell membrane. And simple diffusion, fat-soluble molecules directly through the phospholipid bilayer. And another type, another mechanism is by osmosis. By osmosis means water molecules just diffuse through the lipid bilayer. It's not a lot, just a fraction. And the number three way of transport is through the integral protein. So the protein which span across the phospholipid bilayer act as a gate so that the solutes from outside the cell can go in to the cell, but this process requires energy, which is ATP. And now we also need to talk about exocytosis and endocytosis. Exocytosis is an active mechanism. Exocytosis means from inside of the cell, moving them out. And endocytosis means taking the substances from outside of the cell into the cell, inwards, endocytosis. And in exocytosis is an active mechanism that moves substance from cytoplasm to outside of the cell. Endocytosis move large particles and microtubules to enter the cells. And we have learned this already. Diffusion, particles move from high area to lower concentration. And in osmosis, it needs to be semi-permeable membrane. And only the solvent molecules are free to move to equalize the concentration. So it is not for everything. It is a semi-permeable membrane. Now let's talk about cytoplasm. Cytoplasm, as we saw from the previous diagrams, it is inside the space inside the plasma membrane. It consists of the fluid base, which is called cytosol, and organelles, small little organs of the cells, and inclusion bodies. As cytosol is made of water ion, electrolytes, protein, which is like a jelly-like structure. And inside the cytosol, all these nucleus, mitochondria are suspended. Now let's talk about contents of the cytoplasm. There are many structures, but let's talk about a few important things. For example, like ribosome, endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria, centriosome, centrioles, nucleus, and of course, there are many others which I have not included. Exocytosis and endocytosis, 3 types. This is endocytosis. Endocytosis means taking something into the cell. And another type of endocytosis, and that is the number 3 type of endocytosis. Let's jump and talk about the organelles of the cell. Let's start with the ribosome. What is ribosome? It is construction of ribosomal RNA and proteins. It is not surrounded by a membrane, and it is the site of protein synthesis. The assembly of protein is called translation. From DNA, translate into protein, and therefore we call that process translation. Endoplasmic reticulum, it is a meshwork or network of inside the cytoplasm. There are 2 types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is a flattened disc, and the ribosomes we talk about attached to the external surface of the rough endoplasmic reticulum, and it is for synthesizing protein synthesis. And smooth endoplasmic reticulum, we call it smooth because there are no ribosome attached, and it synthesizes the lipid. So now let's look at that. Now we can see the small little ribosome attached to this endoplasmic reticulum, and therefore this is the rough endoplasmic reticulum. Now look at the similar structure, but there are no ribosome attached, and therefore we call it smooth endoplasmic reticulum. Rough endoplasmic reticulum, with the help of ribosome, it is for protein synthesis translation, and smooth endoplasmic reticulum, it is for lipid synthesis. Golgi apparatus. Its function is for packaging and shipping. It's just like that from a manufacturing plant, you know. This is the Golgi apparatus. It is what it looks like. It is like a sheath, multiple layer. Look at that. So the job of the Golgi apparatus is for packaging, which means those content from the endoplasmic reticulum are coated in the Golgi apparatus, and then they will be secreted or released from the cell. It is very closely related to the endoplasmic reticulum. Mitochondria, that is the energy house. It is one of the most complex organelles. It is responsible for aerobic phosphorylation of ADP, adenosine diphosphate, to ATP, adenosine triphosphate. So it is the energy house. It has outer and inner membrane. Let's look at that. It has outer membrane, inner membrane. It has got crystals inside here. It just looks like a peanut. These are the sequence of events. As you see, ref endoplasmic reticulum, we have ribosome, protein synthesis, they have to go through the Golgi apparatus for packaging, and finally the packed protein will be released from the cell. Lysosome, these are the vesicles which are responsible for containing or sequestering the digestive enzyme, which is to be used by the cell on materials following like pinocytosis or phagocytosis. It contains digestive enzyme, it contains acids, it breaks down the ingested substance. Let's look at these cytoskeleton, which will be microfilaments. These are the thin protein filaments often made from actin subunits. They are the structure which scaffold the cell membrane. And they are the functional proteins responsible for the maintenance of the shape of the cell, depending on internal or external pressure. So there are three types: microfilaments, intermediate filaments, and microtubules. You can see the different size, 7 nanometer, 10 nanometer, and 25 nanometer, and different shapes. These are made from tough protein fibers such as keratin, but let's say it is the actin subunit. So now come to centrosomes and centrioles. Centrosome is a spherical structure in the cytoplasm that organize microtubules. So it composes of matrix and two centrioles. It participates in the formation of cilia, and it is also necessary for nuclear division. Centrioles are the proteins, base organelles. They are only functional during the mitosis, and that is responsible for segregation of chromosome between the daughter cells. They are paired cylindrical bodies. Small little structures like cytoplasmic inclusion. These are the temporary structures. They are not present in all cells. They may consist of pigments, proteins, fat, glycosome, and that sort of stuff. Now we come to the nucleus, which is the control center of the cell. It is responsible for housing the genetic material, such as DNA, messenger RNA, transfer RNA, ribosomal RNA of the cells. It's just like 5 micrometer in diameter. So this is the nucleus, which is surrounded by the rough endoplasmic reticulum, and in the center we have a small little nucleolus. Nuclear envelope, 2 parallel membranes separated by the fluid phase space. and it's continuous with the rough endoplasmic reticulum. There are little pores which penetrate the nuclear envelope, and it allows these little pores, we allow the larger molecule to pass in and out of the nucleus. Nucleolus, it is a darkly stained structure in the center, as we saw. It is the site of ribosome subunit assembly. Okay, finally, here comes to the end of the lecture. We saw a cell, a human being is made of many different cells. We have talked about the boundary of the cell, which is plasma membrane. We have talked about inside of the cell, which contain cytosol and organelles. And if we combine cells, and a cell is a structural unit of living, and if we combine cells with the same function, we get tissue. There are 4 types of tissues in our body, which are epithelial, connective, muscle, and nervous tissue. But essentially, a human being is made of multiple cells. And finally, from the cellular level to we get tissue level, from tissue level to organ level, from organ level to system level, and finally, which we become a human being, a person. Thank you. That's the end of the lecture.